So here we go. Once again, I Ajay Jha on behalf of ACS India team. Welcome you to today's ACS Science Talk session. Just to update you on the upcoming ACS Science Talk, please visit our ACS in India webpage and register for the upcoming session by Professor Aarti Jayaraman on 17th September. We have also updated the past lecture recordings on the ACS Science Talk virtual library. Please feel free to visit, like, subscribe, and share among the scientific community. To keep you updated with the latest research findings, the product launches and services at ACS, we have a monthly newsletter called ACS Insights India. Subscribe today for free. We'll provide the link into the chat box later. To begin with the scientific session, it's a pleasure and honor to have Professor Teri Odom with us. Professor Odom is Charles E. and MIH Morrison Professor of Chemistry and Chair of the Chemistry Department at Northwestern University. She received her BS in Chemistry from Stanford University and PhD in Chemical Physics from Harvard University. Professor Odom carried out postdoctoral work at Harvard University before starting her career at Northwestern University in 2002. She's an expert in designing structured nanoscale materials that exhibit extraordinary size and shape-dependent optical and physical properties. She's a recipient of numerous awards, including the ACS National Award in Surface Science, a Research Corporation TREE Award, the Associated Student Government Faculty Honor Roll, the Carroll Taylor Award from the International Precious Metals Institute, a DuPont Young Investigator Grant, a NSF Career Award, and many more. Professor Odom was founding chair of the Noble Metal Nanoparticles Gordon Research Conference and founding vice chair of Lasers in Micro Nano Biosystems. She was the founding executive editor of ACS Photonics and currently serves as the editor-in-chief of Nanoletters. Her scientific journey has been featured by ACS publications as Odom's personal story of discovery. Now, I would like to invite Professor Odom to deliver her talk.
Thanks, Ajay, and the ACS team. Thank you for inviting me to tell you a little bit about the work that my group does, as well as where we want nanoletters to go in the future. So I assumed the chief editorship of Nanoletters in starting of 2020. And so we've had a 20-year run of being a leader, one of the first nanoscience journals. And now we aim to continue that leadership and making sure that we focus on the communications piece, meaning the tagline is the global home for communications and nanoscience and nanotechnology. So we're an international journal, but we also want to focus on letters. So nanoletters, we have expanded the scope of the types of topics that we're interested in. In the first 20 years, it was mostly focused on fundamental advances, but now we're interested in more applied research as well as emerging research. The other thing that's quite interesting about nano is it's had some time to mature over 20 years. And so we want to ensure that the topics are an intersection of two or more areas. So not just a single area in, say, chemistry. So as I mentioned, we have a communication. We only have communications and no articles, which are longer pieces. I introduced this idea of what's called a viewpoint. It's meant to be some 20-year out musings of some young leaders in nanoscience, emerging leaders, as well as very established pioneers in the field. We've had several Nobel Prize winners write these viewpoints, and just to have some musings of what they think about is possible for their field, as well as nano in general. It's been pretty exciting to read them. I encourage you to do so. We were also one of the first journals to have an early career board. And so this early career board is responsible for helping us promote content on social media, identifying areas that they would like to see as virtual issues, and then writing a piece about that. But also they're the ambassadors for the field in some ways. They're up and coming leaders, and we want to involve them as much as we can. New for 2021, we have an executive editor, E. Sui, and so he is one of the he's still an associate editor, but he helps me with the leadership of the journal. And one of the key leadership roles that he played during this pandemic is to find a way to continue to make connections and to provide content to the entire scientific community. So this is an advantage. So one positive outcome of the continuing pandemic is really the ability to access content unfettered. So you might not have an opportunity to go to a conference in, say, England, but if someone like Andrew Geim can give a lecture within this Nanoscience Global Lecture Series, then this is now available for anyone's consumption. I think this is a huge benefit for students, but also it makes the case for how do we transmit information and key ideas much more efficiently so that more people can be educated and then take that into their own research. So I think that's been very exciting. So we have had around 9 global nanoscience global lectures in 2020. These are all recorded, so you can go back and access them if you're interested in these ideas. We've typically featured 2 senior people and a top emerging junior or just slightly younger junior person. And also within these nanoscience global lecture series, for example, in this November one, all of these 3 speakers contributed viewpoints to Nanoletters related to some of the talks that content of the talks that they discussed. So I encourage you to read them. The viewpoints are very short. They're about 1 to 2 pages, but the articles and their perspectives is very, very impressive. This is the associate editor team for this year. We have broad representation across a wide range of fields as well as countries, and we've hired several new associate editors to cover emerging areas that we've been interested in. So if you're interested in learning more about this, we can talk about that after the scientific talk. So today I want to describe a project that is based in fundamentals, but also has downstream applications. And I think this is how I've been thinking about nanoscience recently. We still have to be very, very good at the fundamentals, because if we don't understand what's happening at the single molecule or atomic level, it's going to be very difficult to translate that into the outcomes that we'd like to see. And so advances in fundamentals, advances in technology transfer or application is, I think, really important for the future of nanoscience. And so I hope to show you a little bit about both of that today in this talk. Also, since this is partnering with India, I thought I'd focus on a general topic from my group that has strengths in India. And I know this just based on some of the papers that we've handled and also some of the collaborative efforts that we've been involved with. But before I get started in the details, I did want to acknowledge and highlight my group, who have been really heroic in this pandemic time. This is a Zoom image of when we were still not really allowed on campus, so the very beginnings of the pandemic. But it's not been easy. It's exacted a toll. It's still exacting a toll, but I always like to be reminded of both the resilience and the optimism that students have. And so for those who are students on this, participating in this video, thank you for the efforts. It hasn't been easy, and we acknowledge your hard work. I'm going to be telling you today about the work of Ting Ting, Emma, and Priscilla in the group. And so I've highlighted their faces with little stars, gold stars. Okay, so one of the fundamental questions that we've been interested in is, how do you place different molecules in specific locations on nanoparticles? And as you might imagine, it's a very challenging problem, not only from the mismatch of scales, but you're trying to take advantage of relative surface energies of the nanoparticle, and then what types of molecules are best to bind, and why would you want to do that? And so just in terms of addressing the first challenge, how do we put different molecules in different spatial locations of a particle? I wanted to highlight some literature at the beginning, because I think there's been very creative approaches to this problem. So one solution to achieving regioselective placement is, especially of DNA, is to take advantage of phase segregation. So what this means is the starting template is a nanoparticle of a different shape, and then a hydrophobic polymer is used to block a portion of it, whether it's a sphere or a portion of a nanorod in different locations, a prism or a cube. And then wherever there's the exposed area, the DNA of its thiolate can be selectively covalently bonded. And so you can see these different transmission electron micrographs. If the polymer blocking layer is here, then you have DNA in all the remaining open areas. So this is a very nice example of the beginnings of regioselective placement. The challenge here is that it's difficult, or it can be difficult, to discriminate between different DNA oligonucleotide sequences. So for example, if we look at this picture of the 2D prism, can you put different DNA molecules at each of these different vertices, or can you mix DNA on the surface? And that's hard for this particular approach. But also in this approach, you have both DNA ligands, and then you have this polymer blocking layer. And so you might imagine that you would like something just with molecules on the surface. And so this has been some of the next advances related to DNA stamping. Again, you start with a bare substrate. There's a DNA frame or origami type structure. You use that for pattern transfer to put different DNA ligands on the surface. And then if the DNA is designed with a complementary sequence, then what is possible is to attach these different ligands with small spheres to indicate that the selective and site-specific assembly has occurred. So this is a very elegant way to achieve selective assembly on these spherical substrates. So the couple of drawbacks here is related to that complex DNA scaffolds are needed. So the design of the scaffold is somewhat complicated, and often it's limited to small spherical particles. So the approach that we have taken is to introduce a new type of scaffold that can only have resultant DNA ligands in specific locations on the surface. And so we like these three-dimensional structures because they show positive curvature, negative curvature, as well as neutral curvature, all in the same structure. And moreover, this local nanoscale structure can give rise to macroscale chemical and physical properties. And a further advantage is that the different curvature regions may affect ligand loading and overall construct properties. So for example, in this positive curvature region, because this is made out of gold, there would be a high local field at this tip region, but also because it's positive curvature, you'd expect the ligands to be displayed. You are able to achieve high packing of the thiolated ligands, for example, in these straight regions or these neutral curvature regions. And if the ligands are assembled in this negative curvature region, you can imagine that there are steric effects, and these steric effects can produce local changes in charge as well as effective pH. So there are a different range of outcomes that is possible depending upon where these ligands are on this three-dimensional scaffold. And my group has focused on this as I mentioned, this is a fundamental problem. How are different ligands able to be assembled on these different structures? But also we've been using these shapes as optical probes to investigate multivalent interactions with single particle probing of protein ligand interactions. So first, let me introduce the synthesis of these types of structures. So we're using a goods buffer, which is used to control the pH in media. And that goods buffer is used as a reducing agent as well as a directing agent of the standard gold salt. And here's an example of how we can scale up the synthesis of these particles. We were interested in the beginning in scalability and grading therapeutic constructs. And you can also see that if we, from the transmission electron micrograph images, that the particles look really quite nice. But you can also see, because it's a room temperature reaction where there are no seed particles, we only have two reagents, that not all the particles are identical. If the goods buffer is changed from heaps to eeps to mops, there are different shapes that result. And these are some reconstructions of these different three-dimensional shapes that are possible. These are electron tomography images and that we've obtained in collaboration with Dr. Sarah Balls. And which is quite amazing that these non-seeded particle synthesis can, just by changing the goods buffer, result in vastly different shapes. And so we can control the optical properties or the localized surface plasmon resonance of these gold nanostars. So if the gold salt concentration stays fixed, we are able to sweep the concentration of the buffer, and then you can see the evolution of the star shape. So what's contributing to the red shifting of this localized surface plasmon resonance is the emergence of some longer branches. And this can be seen also if we compare the heaps buffer versus the eeps buffer. What's interesting about the eeps buffer is if you look at the molecular diagram, that there's just an insertion of a C2 group between the piperazine ring and the sulfonate group. So just one carbon, which is quite different, but then you can see already as you fix the concentration of the eeps and sweep the, of the gold salt and then sweep the goods buffer concentration, you'll see very different shapes that result from these stars. Here's a direct comparison of why the final shapes are so different. So again, there's just an extra carbon in the insertion on the hydrocarbon chain, but you can see for the eeps particles, their branches grow in a specific direction. And for eeps particles, they also grow in a specific direction, which is quite different. So the local effects, you can see that why it's so important to be able to understand at the single molecule, almost at the atomic level, what is necessary and what controls particle growth. We can also see some different effects based on the third goods buffer, which is this MOPS buffer. So the sulfonate group still remains, but instead of the piperazine ring, there's a morpholine ring, and there are two resonances that result as the ratio of goods buffer to gold salt is swept. And that's mostly because, if you can see this here, there's a longer branch, which corresponds to the longer wavelength resonance, and a shorter branch, which corresponds to the shorter wavelength resonance. And these also grow in different directions compared to the heaps. This grows along the similar direction as the eeps particles. And these tend to be a little bit more unstable, but we have ways to post-process the particles using density gradient centrifugation by setting up a linear gradient, and then we can extract different fractions to achieve a higher purity in population or less inhomogeneity in the particle shape. So if we're using these as scaffolds, the first thing we need to do is to be able to ensure that we know how much oligonucleotide is on the surface of the particle. And this is just globally overall for now. And so we used pH to be able to tune the number of strands, whether it was single-stranded DNA, an aptamer against HER2, called these aptamers can be considered, DNA aptamers can be considered chemical antibodies, whether it's double-stranded siRNA or these folded G quadruplexes. We can control the loading of them just by controlling the pH. Using the citrate buffer method, we can reduce the loading process from 2 days to about 10 minutes, which is in advance and the amount of time you need to wait to be able to synthesize the particles, but also the amount of excess DNA that we needed to use was reduced by 1/3. So from a practical consideration, the citrate buffer method is quite good. But we also needed to determine not just quantitatively how many ligands are on the surface, but we needed to ensure that they maintained their shape once they were on the particle versus in free solution. And this is not easy, and hardly anyone does this in the literature because it's a big, it's not that simple. You need a lot of particles to scale up so that you can see the ligand effects on these particles. But we did it because we're interested in understanding, again, at the fundamental level, how many molecules do we have on the surface? And then do they fold in the same way once they're on the surface, so that if we use them for downstream biological applications, we can interpret the results. And we can see that the duplex structure is maintained, as well as these G quadruplex structures are maintained for the different aptamers at high enough pH. So we've been interested in, as I mentioned, using these constructs to probe interactions with cells at the single particle level. So we looked at a couple of different systems. So we have an aptamer, which is, we focused on DNA for stability, which acts like a drug. And then we are interested in how this aptamer binds and interacts with biomarkers that are overexpressed on cells. And so this is the way that we design the constructs. Again, this is just the cartoon version of what I had shown you early on the previous slide, where we had quantitative information. And so if we're interested in membrane expressed interactions, we can design this with the ligand that binds to HER2, and then it follows the endocytotic pathway after receptor mediated endocytosis. We can also bind proteins targets within the endosomes. You can see this with this red antibody, but actually represents a protein. And then looking at these interactions in the endosomes. And another subcellular organelle that we can target is related to the nucleus. And this is nucleolin, being able to bind nucleolin on the particle surface, and then take that, use that construct to deliver drugs at the site of the nucleus. And I want to introduce this latter example first because it illustrates why we care about light triggered release. So I motivated the talk by saying we want to know specifically where are the ligands on the surface and how can we control it. And this is an example of why that is really important. So for example, we've designed these constructs that combine to nucleolin on the surface, and then it's taken up in some way to the cells and then delivered to the nucleus. So you can see in these transmission electron micrographs that where the particles are located near the nucleus, it introduces some change in phenotype. There are these invaginations based on the ligands that the particles have transported to the nucleus. But these types of phenotypes didn't give us as much of a response as we wished biologically, and I'll show you that in a minute. And so we were wondering, well, it's the ligands that are doing the work. Can we take these constructs, subject them to femtosecond pulses of light, and then release the ligands from the surface? And the answer is yes, but where the ligands come off as you might expect or anticipate, is where the fields, local optical fields, are tightly localized, which is at the tips. And so then you can see when you start to apply light to these particles, the phenotype changes dramatically. And these assays, this caspase-3,7 activity gives us information on late-stage apoptosis. So there is activity just based on having the particles close to the nucleus, but you can see that the activity really increases once we've released a portion of the ligands from the particle surface. And this can also be observed if we compare the double-stranded DNA breaks in the nucleus. So all of these green regions here are these DNA damage within the nucleus, and so this is the mechanism by which the particles undergo apoptosis based on these DNA breaks. And so this is quite interesting, and you can see we have been able to see the value of delivering a multi-structural construct, but then taking advantage of being able to selectively release molecules at specific regions intracellularly. Another example of this is to target receptors within the endosome. And the reason that this is important is because it affects some other downstream processes that are key for immunotherapy. So, which takes advantage of your immune system to be able to treat the disease. So how do we stimulate our immune system to be able to solve this problem? So there are a range of immunomodulatory agents such as antibodies, proteins, oligodeoxynucleotides, which we'll refer to as ODNs. And the advance of ODNs is that they show programmable immune activity just based on sequence. And DNA synthesizers have improved quite a bit, so it's quite straightforward to program this in if we understand how it works. And so the immunomodulatory agent that we'll focus on is this ODM with cytosine, phosphate, guanine, CpG motifs. So these motifs are methylated. You can see it right here highlighted in red in mammalian DNA. But if it's unmethylated, then your body, the immune cells will identify any of these markers as pathogenic. And so what CpG, these motifs can do is they combine to toll-like receptors, and these are in the endosomes. And once they're bound to these toll-like receptors, this activation releases cytokines, which can stimulate the immune response. And so this is an example of the process. So the nanoparticles functionalized with CpG can be taken up. They target TLR9 and the endosomes. There's NF-kappa B activation, and then eventually there's cytokine secretion. But what was interesting as we first started investigating this with spheres is that only a small percentage of CpG is actually needed for activation. And that was sort of surprising. And in fact, we only needed as low as 5%. So we put the same number of ligands on these small spherical particles. We just changed the percent of CpG. The other was a control sequence, GPC, which just inverts the C and the G locations relative to the 5' and 3' positions on the DNA. And but we only needed about, if you look at the green, you only needed about 5% CpG in order to induce this immune response. So then we asked ourselves this question, well, can the structural valency of the gold nanostar construct produce an immunostimulatory response that was similar to the spheres? And these are not to scale, but I should have mentioned that these tips end with a radius of curvature of a couple of nanometers, which is the same size as a protein. And so the idea is, can this type of structural valency, if we just have these CpG ligands at the tips cannot produce similar responses of spheres. And so the first step we needed to design is actually, can we control where these DNA ligands are at specific locations on this construct? So we first tested with a very easy sequence, and we just labeled this DNA1, it's poly-T. So we functionalize uniformly on the particle surface. We subject the constructs to femtosecond pulse light, and we expect selective release at the tips that correspond to the wavelength of the light that we're using. Meaning if the localized surface plasmon resonance at 820, you can see in these calculations, the fields are tightly localized when the polarization is along the XY direction, along this X direction. You can see that the fields are tightly localized here, and so you would expect DNA release at these particular positions. And this is the localized surface plasmon for the solution. Okay, so can we get DNA release using this approach? So we tested two different types of parameters. We tested the powers that were needed, as well as irradiation times. And this is, again, just this poly-T sequence. So if we just evaluate the 4 seconds first, so 4 seconds at 0.3 watts per square centimeter, 0.5, 0.7, 0.9, 1.5, and so you can see that as the power is increased, more DNA is released from the particle surface. And as you might expect, if the samples are irradiated for longer times, again, you get selective increase of from the surface. So this is quite nice. Okay, we have 2 knobs to tune in terms of DNA release. But what we had to be careful of is you can keep irradiating, and I showed you up to about 1.5 watts per square centimeter, but you'll notice that we start to observe some of these other features at different wavelengths. And you can see this effect through this sequence of images. So with no irradiation, you see all of these are star-like shapes. At 0.3 watts, you see these are all also star-like shapes. At 0.5 watts, if you squint a little bit, you can start to see a feature around 520, and these correspond to some of the spheres. 0.7 watts, similar. 0.9 watts, you still have some of these star structures, but then more spheres are formed. And this localized surface plasmon resonance at this particular wavelength corresponds to spheres. So if you see it increase, you know that you have spheres within your population of stars. At 1.5 watts, you'll see you have a mix of stars and spheres. And by the time we increase the power to 2.1 watts per square centimeter, the majority is spheres, although we do have some, shorter powers. Shorter irradiation times, we still have some stars, but at longer irradiation times, everything basically becomes spheres. So there's a balance on what we can understand in terms of selective release from the surface. So that was with DNA, a type of DNA that was uniformly around the particle surface. And now we want to put a different type of DNA on the bare part of this nanoparticle. So again, we take this construct, we've exposed regions where now, in principle, a second DNA can bind. And so we found out a couple of things. The first is that dense functionalization of the first DNA sequence is required. So if we didn't have dense functionalization before we started this process, then we will have non-specific additions that would just insert in locations that we couldn't control. So that was important. The second is that the amount of DNA2 added, the number of strands was very similar to the number of strands that were released. And so I should mention that these DNA sequences are of similar lengths. And so the charge is a little bit different, but that didn't play as big of a role as we expected. Okay, so you can see here that after 4 seconds, and then doing this release and then adding the second strands of DNA, we increasingly get DNA 2 addition. So we can quantify the numbers of DNA on the surface. You'll notice that the label says, well, both on the axes and in the scheme, Psi3. So these are all measurements based on fluorescence. So we want to know, we characterize the fluorescence, how many fluorescent DNA ligands are now on this construct. The other item that we found is that salt aging is required during this second functionalization step, meaning as we're adding DNA 2, if we don't provide a way to reduce the repulsion between DNA ligands, then we would hardly get any addition of this second DNA sequence. And so this is pretty neat. So this construct here now has 2 different types of DNA. It's all DNA. No polymer blockings, and in particular, it's at a specific location. So this was a major advance, and we were very happy with this. But the other point in question is that we wanted to know, one, is the DNA functional? Because we don't know that, we've just quantified the DNA. And two, how can we be sure that the DNA is where we want? I mean, we think we have an idea based on what we've done today, but we needed to do one more step. And so we decided to use duplex hybridization based on an intermediate DNA strand to indicate that, yes, we indeed have function, and we indeed have placement as anticipated. So this is the DNA sequence that we added after the initial release. We have this linker strand in the middle, and then we're going to visualize the placement using gold spheres. But again, this is selective based on DNA hybridization. So we are testing the functionality of the newly attached DNA. We said, great, we released the DNA 1, we added DNA 2, is it functional? And 2, we can exploit the high atomic number contrast of the gold spheres since they're electron dense. And so this is the control experiment where we expect gold spheres everywhere around the starting construct. And this is pretty neat. It's an example of affirming that, yes, the new DNA that we added is functional, and 2, moreover, that you can visualize it in this really nice way in these assemblies. Okay, so that was the control. Now we want to know, can we put the ligands where we want in specific locations? So again, we have DNA 1 on the body, and we're looking to see if we have DNA 2 at the tips. And you can see in these series of images that, yes, they're mostly at the tips of these different constructs. I should mention that it's not on all of the tips. And the reason is that these particles are, well, these branches are subject to polarization. The localized surface plasmons depend on polarization. And it turns out that the pump is linearly polarized. So you're only going to be able to excite or release DNA at specific, on specific branches at the same time, which is why there's some asymmetry there. Okay, so that was the case where, whoops, I'm sorry, where we have this DNA 2 at the tips, but we also want to do the reverse, where we have the DNA 2 on the body. And so you can see indeed again that there are no spheres at the tips, but they're all now around the body. And so this is some statistics of this DNA placement. For the most part, we have very good control over where we want the DNA to go, and we can visualize the function and the placement using these smaller 5 nanometer spheres. I think this is actually quite neat. Okay, so in summary, I told you a new strategy to be able to put DNA ligands or any type of thiolated ligand at specific locations on these anisotropic scaffolds. And the second functionalization can create a construct with 2 or more types of DNA on the surface, which I think is a big advance. But moreover, this approach can be used to design new classes of gold nanoparticle superstructures. So our next stages are related to, once we just put these CpG on the tips, then we can test these ideas of whether these enough CpG at these small tips can be compared to result in the immunomodulatory activity that we would anticipate. And so I just want to close the talk with a picture of my group from about a month ago. And this was our first, it was sort of a celebration since students were defending and leaving. So our first picture outside with everybody vaccinated. So it was very exciting. So, so I'll stop there and then be happy to answer any questions that you might have.